Good morning, everybody. Myself, S. Ramachandran, Associate Professor, Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Sciences, Vistas, Chennai, is going to give a seminar on the topic enzymes. The importance of enzymes are they play an important role in metabolism, diagnosis, and therapeutics. All biochemical reactions are enzyme catalyzed in the living organism. The level of enzyme in the blood are of diagnostic importance. Example, it is a good indicator in disease such as myocardial infarction. Enzymes can be therapeutically used, such as digestive enzymes. So what is the definition of enzyme? They are proteins that increase the rate of the reaction by lowering the energy of activation. They catalyze nearly all the chemical reactions taking place in the cells of the body. They are not altered or consumed during the reaction, and they are reusable. So, what are the active sites? Enzyme molecules contain a specific or a special pocket or cleft called the active site, where the substrate can and bind and form the product, which comes in the lock and key mechanisms later on. So this is the substrate which joins with the enzyme. So that is the place where the substrate join is called the active site. The structure of the enzymes is classified into two types. It's called the complex or the bioenzymes and the simple only protein. The complex or the holoenzymes or the bioenzymes or the protein part and the non-protein part is called apoenzyme and the cofactor. The cofactor is subdivided into two types, the prosthetic group and the coenzymes. So coming on to the mechanism of action of enzymes, they increase the reaction rate by decreasing the activation energy, and they form by the enzyme substrate interaction, that is, formation of enzyme substrate complex by the lock and key mechanism or by the induced fit model. You can hear a typical diagram where the energy of activation is lowered, in which the first one there is taking place without the enzyme and the second one with the enzymes. A classic graphical representation of the same processes. So what is this lock and key mechanism? In the lock and key model of the enzyme action, the active site has a rigid shape. Only substrate with a matching shape can fit, and the substrate is a key that fits the lock of the active site. This is an older model, however, and does not work for all enzymes. So what is the induced fit model? In the induced fit model of enzyme action, the active site is flexible and not rigid. The shape of the enzyme's active site and substrate adjusts to the maximum to fit, which improves the catalysis, and there is a greater range of substrate specificity. This model is more consistent with a wide range of enzymes. So you can see there, the shape of the substrate gets altered with the shape of the enzyme. Therefore, it's called induced fit model. A classic example of the lock and key mechanism. So what are the after effects or the affection or affecting factors of enzyme activities? There are three factors. One is environmental conditions, cofactors and coenzymes, and enzyme inhibitors. So what are the environmental conditions? They are the extreme temperature, pH, and the substrate concentration. Second factor is the cofactor and coenzymes. Inorganic substances, zinc and iron and vitamins respectively are sometimes used for proper enzymatic activity. For example, the hemoglobin present in the blood is used for the picking up oxygen, and the iron must be present in the quaternary structure of the hemoglobin. Therefore, oxygen can also be taken place or can be picked up. Environmental factors: optimum temperature, temperature at which maximum activity or reaction occurs at the fastest level. Environmental factors, one more, pH, also affect the rate of reaction enzyme substrate complex. So coming on to the last one, which is nothing but a reversible competitive inhibition. So there is inhibition of the enzyme activity either by two paths, but is competitive and non-competitive. So what is a competitive inhibitor? Has a structure just like the substrate. It complex with sites for the active site, and its effects reversed by increase the substrate concentration. Then what is a non-competitive inhibition? Name is there as a structure different than the substrate, distorts the shape of the enzyme which alters the shape of the active site, prevents the binding of the substrate, cannot have its effect reversed by adding more substrate. So coming on to the very important part, which is called the name of the enzyme. As you all know, ASC is the last term which denotes the enzyme. Like sucrase, oxidase, all these enzymes are there. Sometimes there are some enzymes which are having the alternative name like nothing but the pepsin and trypsin, which are used for the digestion enzymes. There are also names which contain nothing but the dehydrogenase or ethanol dehydrogenase or transaminase. All the other types of enzyme naming. The classification of enzyme is based upon the IUB, nothing but the International Union of Biochemistry, on the basis of type of the reaction that they catalyze. They are catalyzed into six types. Number one is oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Oxidoreductases, nothing but both oxidation reaction, reduction reaction takes place. Transferases, it transfers one group from one to another. Hydrolysis, it hydrolyzes the compounds. Lyases, the same hydrolyze but taken by some other enzyme. Isomerase is nothing but changing from one form to another, and ligase is nothing but the combination of two substrates. So, an example of an oxidation reduction, nothing but your transferases and hydrolysis is present over here. Example of a lyases, isomerases, and ligases is given in this slide. These are the references that I use for my study or my presentation. Thank you.